So I was very flattered when Mark asked me, since Brauman's not here, to introduce our next speaker, Alanna Schepartz. Alanna is a Harris Professor of Chemistry at Yale University. She got her undergraduate degree at SUNY Albany and then moved to work for one of the greats in bioorganic chemistry, Ron Breslow. And from there she moved to another great scientist, Peter Durbin at Caltech. And now she is a great scientist, and she has been given many awards over the course of her career. The Dreyfus Award she won in 1993, and she's been won many American Chemical Society awards, including the Biomimetic Award sponsored by Ron Breslow, and most recently the ACS Award from the Division of Biological Chemistry, which I am now chair of, and the journal itself that Laura Kiesling is the editor of. And she's made really seminal contributions over these years, not only to science, which I'll very briefly speak about, but also to the scientific community as associate editor of JACS, and also starting the first NSF workshop in chemical biology. And her focus over these years has really used a combination of both chemistry and biology. She's been able to synthesize what she calls miniature proteins, where she can build in different binding features, and she can use these miniature proteins to reprogram signaling inside cells. She's been able to pioneer the use of beta peptides to make all kinds of really interesting small protein bundles that have novel chemical functions. And today what she's going to talk to us about, I judge from the title, it must be epidermal growth factor receptor and interaction with EFG. So, Alanna.
So thank you, Joanne. I really would like to start just by thanking the Camille and Henry Dreyfus Foundation. You know, I remember I'm the class of 1993, and at that point I had been at Yale for about 5 years. And as all of you know, that's about the point in your career where hopefully you're beginning to get the sense that you're actually going to be around for a while. But I think more seriously, it's also a very important point career-wise, because it's the time where you can really look critically at your own research program and think about the ways that you want to change it and grow in that program in ways that are more feasible after that wonderful transition that occurs somewhere around year 6. And I have to say that the Dreyfus Award arrived at exactly that time and really allowed me to begin to work on some problems that were a little riskier, a little longer term, a little more sophisticated than the things I had done before. And I'll always be grateful for that, because I can look back now on the changes and decisions that I made in '93 about my own research program and see how those decisions have really kind of helped us make better decisions along the way. So I'll always be grateful for that. So as Joanne mentioned in her really nice introduction, our lab for a long time has been interested in interactions between proteins, their chemistry, their specificity, and the role that those interactions play in biology. And we've made molecules that inhibit interactions between proteins. We've made other molecules that assemble into species that look like and behave like biological macromolecules. But today, I actually want to tell you about a molecule that we didn't make. It's a molecule that Roger Chen made, but we've found some really interesting uses for it. And one of the use that I'll describe today is a way to study protein-protein interactions as they occur in cells, and specifically related to the question of how chemical information is transferred through the plasma membrane. Okay, so this is really a question in information theory. And so irrespective of field, information theory involves a message, right, which originates at a source. It proceeds through a channel and then is received at the other end. And this channel can be lots of different things. It can be a group of 7-year-olds who are talking to each other and playing telephone. It can be a real telephone network, in fact, and that's where information theory first originated. For the purposes of this talk, The channel is going to be a receptor tyrosine kinase, a transmembrane protein that Joanne referred to, called the epidermal growth factor receptor. So for many of you, when you think about how information can make it through this barrier around a cell called the plasma membrane, for some of you, you think that's a really easy problem. Okay, so some of you, those of you who are interested in organic chemistry, will think about natural products or molecules that abide by Lipinski's rules, small cell permeable molecules. And what you realize is this is an easy question, because all the molecule really needs to do is traffic passively through the cell membrane to initiate some biological event inside the cell. And if all biology functioned that way, right, the system, it would be an easy problem to solve. But it doesn't. Because it turns out that there are many molecules that function in biology. I show two here, the epidermal growth factor and the transforming growth factor alpha. These are two proteins that weren't around when Lipinski's rules came down from the sky on a tablet. You know, they're proteins. They don't get into cells by passive diffusion. But somehow, by virtue of a binding event to the very same cell surface receptor, EGFR, they mediate biology inside the cell, right? They lead to conformational changes and protein-protein interactions that lead to a signaling pathway initiated by a kinase. And what's really amazing is that those signaling pathways are not the same. And so somehow, this receptor tyrosine kinase, known as epidermal growth factor receptor, or EGFR, knows whether or not EGF or TGF-alpha is bound on the outside of the cell and makes different decisions, leads to different outputs depending on that information. And so what we became interested in is using a chemical probe to try to understand the differences in the structure of these receptor tyrosine kinases that transmitted that information. And that's the story that I'm going to tell you about today. Okay, so if we just go back to what you would read about in your cell biology textbook, this would be a cartoon that represents the structure of EGFR. So this is the cell membrane. There's a region of the protein called the extracellular region that is exactly what its name suggests. It's on the outside of the cell. There's a single pass transmembrane helix. There's a region called the juxtamembrane segment that we're going to be talking a lot about. And then there's really the business end of the receptor, which is the kinase domain. And what you would read in your cell biology textbook is that these molecules are floating around on the surfaces of cells in their monomeric state. When they bind to EGF or some other ligand, they dimerize. And what you would read in your textbook is that that dimerization event brings one kinase into close proximity of the other kinase, and that increased proximity makes the close by kinase a better substrate. You get phosphorylation and signaling occurs. So that model explains, right, how a ligand can lead to kinase activation. But of course, it doesn't explain how the kinase knows which ligand is bound on the outside of the cell. And in fact, it's even more complicated because this model's not really right. And there's another intermediate that I have to say with pride that was identified by my colleagues Derek Toomray and Yossi Schlesinger at Yale. There's this other intermediate that's really an inactive dimer. So floating around on your cell surface are not just inactive monomers of EGFR, but dimers of EGFR that happen to be inactive. Now, it's still true that EGF or a ligand binds and leads to a signaling event. But now, that really can't be due simply to proximity, because in this inactive dimer, the two kinases are at some level of approximation still close. So it doesn't explain that. Okay, it also doesn't explain something that is a little bit of a subtlety, but is important, which is that structural biology experiments have shown that this dimer, this active dimer, is asymmetric. So the C-terminal lobe of one kinase interacts with the N-terminal lobe of the other. And so this model doesn't explain how that asymmetric interface is established. And finally, it says nothing about how ligand identity is communicated. Okay, so there's some information about how ligand identity is communicated. And it turns out that this region that I pointed out earlier, this juxtamembrane segment, this relatively unstructured region between the end of the transmembrane helix and the beginning of the kinase domain, seems to be important in, one, knowing whether anything is bound, Okay? But whether or not it's involved in what ligand is bound has not been established. So what's known is that this juxtamembrane segment is a 37 amino acid sequence, right, that seems to regulate the transition between an inactive dimer to an active dimer. So if you eliminate this juxtamembrane segment, the kinase no longer signals. Phosphorylation doesn't occur. And it also turns out that if you make very subtle mutations in this juxtamembrane segment, it abolishes a subtle feature of EGFR biochemistry, which is that the first EGF seems to bind with much higher affinity than the second EGF seems to bind. So that negative cooperativity, which is a matter of current controversy, right, is eliminated when you make small changes in the juxtamembrane segment. So it's clear that this is an important region. The only other piece of information that was known about the juxtamembrane segment before the experiments that I'll describe to you is some beautiful work that came out of John Currien's lab at Berkeley. And so what his lab did is they looked at the sequence of the juxtamembrane segment and synthesized a 15 amino acid peptide from the N-terminal region of that segment. And they placed that synthetic peptide in an NMR tube. And what those experiments revealed is that that sequence of amino acids has the propensity to exist as a helix. So the NMR showed long-range NOEs that were consistent with helical structure, but were not plentiful enough to allow them to generate a structure. What they then did is simply synthesize a dimer of that peptide, where you have two copies linked by a very simple 5 amino acid spacer, and place that synthetic peptide into an NMR tube. And lo and behold, when you've enforced a dimer by virtue of this very short peptide sequence, now they have enough NOEs with some computational modeling to generate this structure, which is an anti-parallel coil-coil, where this sequence forms a helix, this sequence forms a helix, and this helix and this helix interact in an opposite orientation, where one is traveling in an N to C direction and the other is traveling in a C to N direction. But of course, so what this experiment showed, right, is that this anti-parallel coil-coil is energetically accessible in vitro. But of course, the presence of that very short linker enforces an anti-parallel arrangement because it's only 5 amino acids. It sheds no light on whether or not this coil-coil forms in intact EGFR receptors in live mammalian cells. It sheds no light on whether or not the coil-coil formation depends on the binding of EGF. And of course, it says nothing about whether or not this coil-coil is related to how the receptor communicates ligand identity. And so that's where our experiments began. But in order to tell you about the experiments we did, I have to tell you about this molecule that Roger Chen made. And so years ago, so this is now 14 years ago, in a beautiful paper in Science, what Chen reported is that this interesting fluorescein derivative, in which 2 of the carbon-hydrogen bonds have been changed to arsenic, had the ability to light up in cells in the presence of proteins that carried 4 cysteines in a very close arrangement in linear sequence. And so if you had cysteine, and then 2 amino acids and another cysteine, then that recombinant protein would bind to this fluorescein derivative in cells and light up to emit green light. A few years later, it was shown that the same thing would be true with a slightly different derivative that emitted in the red, which happens to be more useful for experiments in cells, and also pointed out that the sequence of amino acids represented by X was really important, and that if it was proline glycine, then the emission would be intense, the quantum yield of that fluorescent complex would be high, and the dissociation constant would be very low. And that if you change that proline glycine to virtually any other sequence that they evaluated, either the brightness of the complex suffered, or the stability of the complex suffered, or both occurred. And so the overall conclusion of those studies was that that the sequence that existed between these 2 pairs of cysteines were absolutely essential. And so what we wondered is whether or not you could replace that intervening sequence with either an entire protein or by 2 proteins. And if you could do that and engineer a system where the cysteines were still close enough in space to form a complex, maybe X could be hundreds of amino acids or 2 separate proteins. And if those 2 protein regions or proteins assembled into a complex that positioned those 4 cysteines in exactly the same orientation that they would be positioned in this short peptide, maybe you could use this molecule as a tool to study conformational changes in cells or protein-protein interactions. And that's the work that Nathan Lutke did in my lab several years ago. And so what he showed is that using this red-shifted version of this bioarsenical dye that's called ReASH, you could differentiate between folded and unfolded small proteins and, in fact, detect the formation of interactions between two coils, which is exactly what Rebecca Scheck proceeded to do in our lab. OK, so Rebecca's project is very simple. We start with epidermal growth factor receptor that has been modified in two ways. It has a tag at the N-terminus that allows us to know which cells are expressing that protein, and that tag glows green. And it also has two cysteines somewhere in the juxtamembrane segment. Right? Since we have a structure, a proposed structure of what that coil looks like, we can place those cysteines in exactly those positions to generate a high-affinity ReASH complex if that coil exists in cells in the presence or absence of EGF. So the experiment is we add EGF or not, we add this bioarsenical dye, and then we look at the cells to see whether or not they grow just green or whether or not they glow green and red. So the first molecules that we made are the two shown here. The first one has two cysteines in exactly the right position for binding ReASH if the coil forms. And the second has two cysteines in position that should bind to ReASH whether the coil forms or not, because they're just at the end of this transmembrane helix. And I just have to make the point that we're doing this in the context of a protein that has 59 additional cysteines, okay, including 9 in the intracellular domain. But since it has no cysteines that are right next to one another, we can see these proteins on the surfaces of cells over whatever background happens to be in the cytosol using turf microscopy, which is what we did. And I just have to point out that our ability to design these cysteines into these coils was aided enormously by an NMR structure that showed how the bioarsenical dye was coordinated to 4 cysteines in the context of a short peptide. OK, so the first experiment is very simple. We take mammalian cells, and they've been transfected with one of the two EGFR constructs that I just showed earlier. We have to inhibit receptor internalization to keep the receptors on the cell surface. We label with ReASH so that we've added no EGF. And then we look to see whether the cells are green. They're green if they're expressing EGFR, or if they're green and red, and then they're expressing EGFR and they bind ReASH. And so in this experiment, we're asking the very specific question of whether or not these EGFR molecules are assembled into dimers in the absence of EGF. The results are shown here. So now, when we're looking at the construct that has the cysteines just at the end of the transmembrane segment, you can see the cells are green and the cells are red. And so these molecules are binding ReASH even in the absence of EGF. But now, when we look at the construct that has the cysteines within the juxtamembrane segment, in exactly the position that they would have to be to bind ReASH if the coil has formed, now what you can see is that this cell glows green, which means it's expressing the receptor, but there's very little or no red fluorescence. And so what that says to us is that this coil is not formed in the absence of EGF. Then all we do is add one more step, where now we treat with EGF. And I think what you can now see is that now not only does the construct with the cysteines at the end of the transmembrane helix glow red, but now the construct with the cysteines within the juxtamembrane segment glows red as well. So if you were a first-year graduate student, you would rush into my office and you would say, we have evidence for the coil. But of course, that's not true, because all this really says is that those two regions of the protein are closer together in the presence of EGF. It doesn't tell you that there's a coil. But the beautiful thing about coils is that they have incredibly predictable structures. And so the molecule, the construct, the EGFR variant that I talked about that glows red is the one in which the cysteines are really at the A and D positions, right at the interface between the two coils. So of course, they have to move apart a little bit to binding the dye. But we could then make other constructs that have the cysteines not at that interface, but moved around so that they're either on one face of that coil dimer or the other face. And our prediction would be that if the Currian coil exists, then all 3 of these molecules should be able to bind ReASH and light up in the presence of EGF. And we're also able to design other constructs in which we move the cysteines around even more and predict that if they're just close, right, but not in that coil, right, these molecules would distinguish that difference, because if the Currian coil is formed in those cells, these molecules should not light up when ReASH is added. And in fact, that's exactly what we observe. And so here is the histogram plot of the data. What we can see is that in the presence, that when we look at CCH1, 2, and 3, which we would predict to bind, to light up with ReASH in the presence of EGF, that's exactly what we observe for CCH1, CCH2, and CCH3. But for CCH4, 5, and 6, that our model would predict should not bind ReASH in the presence of EGF, we see no increase in ReASH fluorescence. Okay, so those experiments tell us that the pattern of ReASH reactivity that we see in the presence of epidermal growth factor is entirely consistent with the modeled structure that came out of the Currian lab. So an anti-parallel coil. Okay, but now the situation gets a little more complicated and a little more interesting, because the fact is that EGFR is activated by a suite of growth factors. One of them is EGF. Another is a molecule, a protein called heparin-binding EGF. And a third is transforming growth factor alpha. And although these two proteins have very similar profiles in terms of gene activation, the profile for transforming growth factor alpha is different. And so we were wondering whether or not the coil or the different coils that one might be able to form from these sequences might contribute to the information flow that identifies which ligand is bound. So the first experiment that we did was with heparin-binding EGF. And what we see is that in the presence of HB EGF, so now the cells have been stimulated not with epidermal growth factor, but with HB EGF, what we see is that just as we found with normal EGF, we see red fluorescence with the construct with the cysteines exactly as they should be if the Currian coil has formed. But the surprise is that when we stimulate the cells not with HB EGF, but with TGF-alpha, we see very little ReASH fluorescence. And in fact, that's true even if we stimulate with a 10-fold excess of TGF-alpha. And so what that experiment tells us is that there may be a structure formed in the presence of TGF-alpha, but it's not the same coil that exists in the presence of epidermal growth factor. So one of the things that I didn't mention when I had all those coils up is that two of those coils, so in some sense the way we designed those new constructs was to take the Currian structure, the Currian antiparallel coil, and either translate the two strands, so move them axially, or rotate them. Okay? And so CCH5 and 6 were the constructs in which they were either translated or rotated. And the interesting thing, what we see, is that when we take cells that have been transfected with CCH5 or 6 and stimulate them with TGF-alpha, now what we observe is that they light up in the presence of ReASH. Okay, all the receptors are active, and now in the presence of TGF-alpha, we see these cells light up in the presence of ReASH. Although as I showed earlier, when cells that are expressing these receptors do not light up when they're stimulated with epidermal growth factor. Okay, so what that says to us is that TGF-alpha results in a conformation within the juxtamembrane segment that is distinct from the one that that is formed in the presence of EGF, that it's a different coil. And we'll talk in a minute about what that coil might be. So now I just want to transgress just for a second and tell you about a really interesting experiment that relates to this. So the experiments that I've described so far relate to information flow from the extracellular domain into the coil. And so what we've seen is that depending on which ligand is bound, you seem to get a different structure within that region of the protein. But that begets the question of what happens between the coil and the kinase domain. And to address that question, we could make use of a variety of mutant EGFRs that were already known in the literature. And so it turns out that a mutant had already been reported in which two of the arginines that are in that coil are changed to glycine. Glycine is a helix destabilizing residue, and so one would anticipate if those two arginines are changed to glycine that it would be more difficult for that coil to form. And indeed, the mutant of EGFR with those two arginine to glycine mutations is inactive. It doesn't signal. Okay, and what you would also expect is that those molecules shouldn't form a coil because they have these two arginine to glycine mutations. So what's known is that when you disrupt the coil, you abolish this asymmetric interface between the two kinases, and the kinase is inactive. There's another mutant that was known, And that's a mutant in which the amino acid change is in the kinase domain itself. So this is the V924R mutation, in which a hydrophobic residue, valine, has been changed to a positively charged residue, arginine. And what that does is disrupt the interface, this asymmetric interface that is required for kinase activation. And so what we wanted to know is whether or not, if you disrupt this asymmetric kinase interface, do you abolish formation of the coil-coil? And in fact, you do. And so what we see here is that when we take cells that are expressing those two different forms of mutated EGFR, even though they all have the cysteines in exactly the right place for binding ReASH if the coil-coil forms, when the juxtamembrane segment has been mutated to contain those glycines, we don't see ReASH-binding. But I think even more interestingly, when the juxtamembrane segment is pristine, okay, it just has those two cysteines, and we install a mutation at the asymmetric interface between the two kinases, now the coil-coil no longer forms, even though this region of the protein is 200 amino acids from where the cysteines would be binding to ReASH. And so what this experiment tells us is that coil-coil formation demands both the ligand binding event and the formation of an asymmetric kinase interface, and I think really established that these three EGFR domains, the extracellular domain, the juxtamembrane segment, and the kinase, are linked through these synergistic conformational changes. Okay, so now we have this model. Okay? So we have an equilibrium between monomers and preformed dimers on the surface. When they bind EGF, they form one coil-coil. When they bind TGF-alpha, they form another coil-coil. And so this actually addresses some interesting interesting previous results in the EGFR field. It explains how this relatively unstructured region of a protein can actually communicate ligand-specific information, and it just has to do with whether or not the fact that it can form two different structures, at least two. And then it begins to explain the fact that EGF and TGF-alpha signal differently, something that we're now investigating with great enthusiasm. But like most, like all interesting projects, right, at the end, when you write the paper, there are more questions that come up than there are answers. And so some of the questions that I will tell you about and others that we're working on are shown on this slide. So all of you, the fact is that EGFR is incredibly well studied, in large part because it's incredibly important in cancer. And so it is either overexpressed or mutated in a large number of cancers. And in fact, there's one specific mutated form of EGFR that's a critical mutation in non-small cell lung cancer, which is the one that we will talk about. And so the question we asked is whether or not we can understand the aberrant signaling of oncogenic EGFR mutants or the way that they respond to tyrosine kinase inhibitors by probing the conformation of this coil-coil in the mutated versions in the presence and absence of different tyrosine kinase inhibitors. Another question you might ask is, okay, well, what is the structure of the coil-coil in the presence of TGF-alpha? We don't have a structure for that, and that's a question that Amy Dorner is probing. One question that we're very interested in, but I won't have time to talk about today, is how do these different coil-coils lead to different signaling? Do they lead to different changes in the phosphorylation of the C-terminal tail? Do they lead to accretion of different factors? Right? Do they lead to different trafficking as these bound receptors are trafficked through the endocytic pathway? Those are all questions that Rebecca Scheck is investigating. And Julie Lancel is very interested in using these engineered receptors to try to identify inhibitors that perhaps even alter signaling along one pathway and not the other. But today, I'm going to focus very briefly on these two questions in the final minutes of this presentation. Okay, so what I show you on this slide are two crystal structures. The one on the left is the crystal structure of the kinase region of the kinase domain of EGFR in what is considered an active conformation. What I show you on the right is the structure of the L858R mutant of EGFR. That's the mutation that's found very frequently in patients with non-small cell lung cancer. Okay, and what you can look at these structures, and if you look at it quickly, you're going to say, I screwed up, and I really pasted the same picture on the same slide. But that's not true, and you can tell by looking at this region. Right? There are small differences in this beta sheet that's on the right of the structure. But other than that, right, they are almost exactly the same. And so that has led to the proposal that what this mutation does is drive formation of the asymmetric kinase interface that leads to an active EGFR. And in fact, what's known is that the EGFR that harbors this 858 mutation is about 50-fold more active than EGFR is and doesn't require any EGF in order to activate and signal down the EGFR pathway. And so that has led to the proposal that this L858R mutation drives formation of the asymmetric interface. Our hypothesis is that what this mutation does is drive formation of the antiparallel coil. And in fact, that's absolutely true. And so if you now take cells that are expressing EGFR that carries the cysteines at the positions that would detect the Curry coil, if they harbor the L858R mutation, we now do the same experiment in the presence or absence of EGF, and we look to see whether or not the coil is formed. And I think what you can clearly see is that that coil exists, as judged by this ReASH-binding data, either in the absence of EGF or in the presence of EGF. And in fact, that's also true for CCH2 and 3, and we're now investigating whether or not that is not the case for CCH4, 5, and 6. Okay, and Melissa is actually now very interested in investigating how changes in this coil structure, how the coil responds to the presence of various tyrosine kinase inhibitors. Okay, but now let's get to the second question. So what is the structure of the coil in the presence of TGF-alpha? So we actually have been investigating that problem from a number of different directions. Some of them are structural biology directions. Those have failed. Some of them are computational experiments followed by experiment, and those have succeeded. So those are the ones I'm going to describe. So Amy Dorner is a terrific new graduate student who is unbelievably good at programming. And so she used Rosetta Doc to ask two very general questions. One is how promiscuous these two helices are. So when this region of the juxtamembrane segment is assembled into a helix, is the antiparallel coil whose structure we know the only low energy structure that could exist? Or are there many different arrangements of those two strands that are relatively similar in energy? And so she asked that question initially using Rosetta Doc. So she generated 25,000 different structures and looked at the top 25 clusters of those structures. And we learned some things that are very interesting. Right? One thing that was interesting is that only a minority of those structures are symmetric. And I have to say that as I think about this more, I'm not sure we made the right decision, but we chose to look only at the symmetric structures for the experiments I've described. We may, in fact, revisit that. And if you look at all of those symmetric structures, what you see is that they fall into three general categories, and not all of them are antiparallel. So about 2/3 of those structures are antiparallel, where the two strands are running in the same direction as they would be in the Curry coil. But about 1/4 of them have the strands running in the same direction, and 11% of them seem to have the helices that are crossed, where they only have a small subset of residues that are interacting. And so the details of this slide notwithstanding, what this really said to us is that these are very promiscuous sequences of amino acids. And there's actually the possibility for a large number of different interactions between them, which is really exciting when you think about the previous studies where we're looking at the effects of tyrosine kinase inhibitors and thinking about the role that those binding events could have on the effect on the overall activation of the kinase receptor. Okay, but if one looks at all of the antiparallel structures, one can see they fall into a variety of different categories. A fraction of them look very much like the Korean antiparallel coil, but that's only 12% of those structures. There are structures that differ simply by a translation of those two helices. There are others that differ by simply a rotation. There are some that differ by a rotation and a translation. And it turns out that if you look at these structures in the context of the data that we already had regarding which geometries of the helices should result in binding to ReASH in the presence of TGF-alpha, what you see is that these two structures, the ones that involve both a rotation and a translation, place the cysteines in such a way in CCH5 and 6 that they should bind ReASH. But the beautiful thing is that they allow us to generate a new construct, CCH9, okay, that now is designed simply on the basis of those computationally generated structures. And what we see now is that when cells are transfected with that new EGFR derivative, Now, when they're treated with either EGF or TGF-alpha, what we see is that those constructs now don't light up in the presence of EGF, but they now light up in the presence of TGF-alpha. So I think we have a much better idea of what the structure of that coil is in the presence of this other growth factor. Okay, so what I've tried to share with you today are a series of experiments that are looking to understand how chemical information is transferred through the plasma membrane. So I think what we've learned is that the coil that forms in that juxtamembrane region is incredibly important. I think what we've seen, especially for today, is that this information was gleaned with a chemical tool, which is great. One of the things that we're really interested in now is to go back to the first slide and think about information theory. Right? Sometimes there's noise. There will be noise in this signaling pathway. And can we use single molecule experiments to try to understand how many times the signal that is outputted at the level of the kinase is not the signal that you would expect based on the ligand that is bound to the outside surface? That's something that we're very interested in looking at. We're also interested in whether or not this information could inform inhibitor design by looking at how the coil structure assembles in the presence of different tyrosine kinase inhibitors. And finally, right, we're interested in taking what we've learned from EGFR and applying it to other receptor tyrosine kinases and other transmembrane proteins that mediate information transfer. And I have to say, I had nothing to do with this slide. This slide is 100% Rebecca Scheck. And it's actually, I think it's a great, it's Rebecca Scheck from its conception to its execution. And so in Rebecca's mind, EGF and TGF-alpha are traveling along a highway. And that highway diverges into two different signaling pathways. And she's trying to represent that when EGF binds, you get this different coil, and when TGF-alpha binds, you get this coil. I thought this was brilliant, and I use it in every talk now. It's just great. All right, so I'd just like to tell you a little bit about some of these people. So the experiments that I described today were really begun by Nathan Lutke, who's now, I think, a senior person at the University of Zurich. He was helped in the end game by Rachel Dexter, a Caltech undergraduate, Dan Freed, a University of SUNY Binghamton undergraduate, and Sarmis Thuray, who's now at the Rockefeller. The experiments I described to you were mostly carried out by Rebecca Scheck, an unbelievably talented postdoc who did her graduate work at Berkeley with Matt Francis. She's on the job market this year, and I hope she does very well. She should. She's been helped by Kevin Bettendorf from St. John University, Amy Dorner from Whitman College, Julie Lancel, another Berkeley undergraduate, and Melissa Lauder from UVA. And they're a wonderful group of students to work with, and I'd like to thank them, and I'd like to thank you, and I'd be happy to take any questions.
Okay, we have time for a couple of questions. Anybody have any questions? Fantastic
Ruben.
talk.
Alanna, that's just beautiful work. I'm thinking about dynamics and particularly how it might relate to transitions between different forms of these cold coils. Have you thought a little bit about how maybe you have conformational equilibrium between many of these different structures and binding of the ligand just shifts that equilibrium or traps a particular state, and how does that relate to noise, so on and so forth?
I actually think that that's the answer, right? I mean, my bet is that these sequences are all very close to each other and they all have very similar energies.
Right.
And we're looking at a very small shift in equilibria that's driven by conformational changes elsewhere in the protein. So one of the things that I didn't describe, just for lack of time, is that we actually did a fair bit of homology modeling to try to think about how conformational changes in the extracellular domain might actually be transmitted through the transmembrane sequence. And it's actually kind of an interesting thing to do because, of course, the structures of the extracellular domain of EGF are bound to EGF and TGF-alpha were published as back-to-back papers in Cell. And if you look at those papers and the discussion, the structures are discussed as if they're identical, but they're not. And so you can do a fair bit of molecular modeling and just looking to predict that there is a small change in the hinge of domain 4 as the two different receptors move into the transmembrane sequence that can change the relative orientation in which the two transmembrane segments interact with one another.
Shift that equilibrium.
And in fact, so Tim Springer has done some really beautiful biochemistry to look at the differences in disulfide cross-linking depending on when EGF is bound. We're now repeating those experiments in the presence of TGF-alpha to provide evidence for our hypothesis that when the two different ligands bind, they lead to a very small change in conformation that is transmitted through the TM into this change in relative crossing angle. And that change in relative crossing angle is then the difference that leads to the change in the coil. So I do believe that these are all very closely related energy states that are simply shifted in one direction to the other. But that begs the question of what the dynamics are.
Sure.
And we're actually, really thinking hard, but we don't have a good answer. It would be wonderful if we could talk together about ways that we could investigate the dynamics with which these two coils interact in the context of a single molecule experiment. That would be fantastic to do.
Any other questions? Okay, I guess we'll move on to posters. We're having another poster session. Thank you so much. It was a great talk.
Thank you.